Hello friends, warm welcome in this video, that is the secondary lymphoid organs. Friends, the lymphoid organs are broadly categorized into three groups, that is primary lymphoid organ, secondary lymphoid organ and tertiary lymphoid organ. Friends, primary lymphocytes are the organs where the lymphocytes and myeloid cells get synthesized and develop to maturity. The two types of primary lymphoid organs are bone marrow and thymus. The T lymphocytes get matured in the thymus, whereas the B cells, monocytes, dendritic cells and granulocytes get matured in the bone marrow. On the contrary, secondary lymphoid organs is the part of body where a microenvironment is created to encounter and evoke immune response against a antigen. Broadly, the secondary lymphoid organs are categorized into two groups. One which is highly organized and second which is less organized. The highly organized secondary lymphoid organs include lymph nodes and spleen, whereas the somewhat less organized system of secondary lymphoid tissue, referred to as mucosa-associated lymphoid tissue or MALT, is found associated with the linings of multiple organ systems, which includes gastrointestinal and respiratory tracts. Mucosal-associated lymphoid tissue include tonsils, Peyer's patches, which are present in the small intestine, and the appendix, as well as numerous lymphoid follicles within the lamina propria of the intestine and in the mucous membrane lining of the upper airways, bronchi and genitourinary tract. Friends, the tertiary lymphoid tissues also organize and maintain an immune response. The tissues that are sites of infection are referred to as tertiary lymphoid tissue. Lymphocytes activated by antigen in secondary lymphoid tissue can return to these tertiary lymphoid tissues, that is lung, liver or brain, as a effector cells and can also reside there as a memory cells. It also appears as if tertiary lymphoid tissue can generate defined microenvironments that organize returning lymphoid cells. Scientists have recently found that the brain establishes reticular system that guide lymphocytes responding to chronic infection with a protozoan that causes toxoplasmosis. So friends, now we will learn the secondary lymphoid organs in detail. Although the secondary lymphoid organs vary in their location and the degree of organization, they share some key features. That is, all the secondary lymphoid organs include anatomically distinct regions of T cells and B cells activity, as well as all develop lymphoid follicles which are highly organized microenvironments that are responsible for the development and selection of B cells that produce high affinity antibodies. Friends, the lymphoid organs are connected to each other and to infected tissue by two different circulatory systems, that is the blood and lymphatic system. The immune cells are the most mobile cells in body and use two different systems to traffic tissue, the blood system and the lymphatic system. The blood has access to the virtually every organ and tissue and is lined by endothelial cells that are very responsive to inflammatory signals. Hematopoietic cells can transit through the blood system away from the heart via active pumping networks, that is arteries, and back to the heart via passive wall based system, that is veins, within minutes. Most lymphocytes enter secondary lymphoid organs via specialized blood vessels and leave via the lymphatic system. The lymphatic system is a network of thin walled vessels that play a major role in immune cell trafficking, including the travel of antigen and antigen presenting cells to secondary lymphoid organs and the exit of lymphocytes from lymph nodes. Lymph vessels are filled with a protein-rich fluid, that is lymph, derived from the fluid component of a blood, that is plasma, that sifts through the thin walls of capillaries into the surrounding tissue. Friends, in an adult, depending on size and activity, seepage can add up to 2.9 liters or more during a 24-hour period. This fluid, called interstitial fluid, permeates all tissue and bathes all cells. If this fluid were not returned to the circulation, the tissue would swell, causing edema that would eventually become life-threatening. We are not afflicted with such a catastrophic edema because much of the fluid is returned to the blood through the walls of venules. The remainder of the interstitial fluid enters the delicate network of primary lymphatic vessels. The walls of the primary vessels consist of a single layer of loosely apposed endothelial cells. The porous architecture of the primary vessels allows fluids and even cells to enter the lymphatic network. Within these vessels, the fluid, now called lymph, flows into a series progressively larger collecting vessels called as lymphatic vessels. All cells and fluids circulating in the lymph are ultimately returned to the blood system. The largest lymphatic vessel, that is the thoracic duct, empties into the left subclavian vein. It collects lymph from all of the body except the right arm and right side of the head. Lymph from these areas is collected into the right lymphatic duct, which drains into the right subclavian vein. By returning fluid lost from the blood, lymphatic system ensures steady state levels of fluid within this circulatory system. Friends, the heart does not pump the lymph through the lymphatic system. Instead, the slow low pressure flow of lymph is achieved by the movements of surrounding muscles. Therefore, activity enhances lymph circulation. Importantly, a series of one-way valves along the lymphatic vessels ensures that lymph flows in only one direction. So now we will learn about the first lymphoid organ, that is the lymph node. The lymph node is highly specialized secondary lymphoid organ. Unlike the spleen, it also regulates red blood cells flow, and in fact, lymph nodes are fully committed to regulating an immune response. They are encapsulated bean-shaped structures that include network of stromal cells packed with lymphocytes, macrophages and dendritic cells. Connected to both blood vessels and lymphatic vessels, lymph nodes are the first organized lymphoid structure to encounter antigen that enter the tissue spaces. The lymph node provides ideal microenvironment for encounters between antigen and lymphocytes and productive organized cellular and humoral immune response. Structurally, a lymph node can be divided into three roughly connective regions: the cortex, the paracortex and the medulla, each of which supports a distinct microenvironment. The outermost layer, the cortex, contains lymphocytes, most of which are B cells, macrophages and follicular dendritic cells arranged in follicles. Beneath the cortex is the paracortex, which is populated largely by T lymphocytes and also contains dendritic cells that migrated from tissue to the node. The medulla is the innermost layer and site where lymphocyte exists, the lymph node, through the outgoing lymphatics. It is most sparsely populated with lymphoid lineage cells, which include plasma cells that are actively secreting antibody molecules. Antigen travels from infected tissue to the cortex of the lymph node via incoming lymphatic vessels, which pierce the capsule of lymph node at numerous sites and empty lymph into the subcapsular sinus. It enters either in a particulate form or processes and presented as a peptide on the surface of migrating antigen presenting cells. Particulate antigen can trapped by resident antigen presenting cells in a subcapsular sinus or cortex, and it can be passed to other antigen presenting cells including B lymphocytes. Friends, alternatively, particulate antigen can be processed and presented as a peptide major histocompatibility cells complexes on cell surfaces of resident dendritic cells that are already in the T cell rich paracortex. The second lymphoid organ is spleen. The spleen organizes the immune response against blood-borne pathogens. The spleen, situated high in the left side of the abdominal cavity, is a large ovoid secondary lymphoid organ that play a major role in mounting immune response to antigens in the bloodstream. Whereas lymph nodes are specialized for encounters between lymphocytes and antigen drained from local tissues, the spleen specializes in filtering blood and trapping blood-borne antigens. Thus, it is particularly important in the response to systemic infections. Unlike the lymph node, the spleen is not supplied by lymphatic vessels. Instead, blood-borne antigens and lymphocytes are carried into the spleen through the splenic artery and out via the splenic vein. The experiments with labeled lymphocytes showed that more recirculating lymphocytes pass daily through the spleen than through all the lymph nodes combined. The spleen is surrounded by a capsule from which a number of projections, that is trabeculae, extend, providing a structural support. Friends, two main microenvironmental compartments can be distinguished in splenic tissue: the red pulp and white pulp, which are separated by specialized region called the marginal zone. The splenic red pulp consists of network of sinusoid populated by red blood cells, macrophages and some lymphocytes. It is the site where old and defective red blood cells are destroyed and removed. Many of the macrophages within the red pulp contain engulfed red blood cells or iron containing pigment from degraded hemoglobin. It is also the site where pathogens first gain access to the lymphoid rich regions of the spleen known as the white pulp. The splenic white pulp surrounds the branches of the splenic artery and consists of the perioarticular lymphoid sheaths populated by T lymphocytes as well as B cell follicles. As in lymph nodes, germinal centers are generated within these follicles during the immune response. The marginal zone which borders the white pulp is populated by unique and specialized macrophages and B cells which are the first line of defense against certain blood-borne pathogens. Friends, blood-borne antigens and lymphocytes enter the spleen through the splenic artery and interact with first cells of marginal zone. In the marginal zone, antigen is trapped and processed by dendritic cells which travels to the perioarticular lymphoid system. Specialized resident marginal zone B cells also bind antigen via complement receptors and convey it to the follicles. Migrating B and T lymphocytes in the red blood enter sinuses in the marginal zone and migrate to the follicles and the perioarticular lymphoid system respectively. Friends, although animals can lead relatively healthy life without a spleen, its loss does have consequences. In children, in particular, splenectomy, that is the surgical removal of a spleen, can lead to overwhelming post-splenectomy infections syndrome characterized by systemic bacterial infections caused by primarily Streptococcus pneumoniae, Neisseria meningitis and Haemophilus influenzae. Although fewer adverse effects are experienced by adults, splenectomy can still lead to an increased vulnerability to blood-borne bacterial infections, underscoring the role of spleen plays in our immune response to pathogens that enter the circulation. It is also important to recognize that the spleen has other functions, for example, in iron metabolism, thrombocyte storage, and hematopoiesis that will also be compromised if it is removed. Friends, lymph nodes and the spleen are not only the organs that develop secondary lymphoid microenvironments. T and B cell zones and lymphoid follicles are also found in mucosal membrane that lie in the digestive, respiratory and urogenital system as well as in the skin. Mucosal membranes have combined surface area of about 400 meter square, which is nearly the size of a basketball court, and are the major sites of entry for most pathogens. These vulnerable membrane surfaces are defended by a group of organized lymphoid tissue which is collectively known as mucosa associated lymphoid tissue, that is MALT. Lymphoid tissue associated with different mucosal areas is sometimes given more specific names. For instance, the respiratory epithelium is referred to as bronchus associated lymphoid tissue or BALT, or nasal associated lymphoid tissue known as NALT, and that associated with the intestinal epithelium is referred to as gut associated lymphoid tissue or GALT. The structure of GALT is well described and ranges from loose, barely organized clusters of lymphoid cells in the lamina propria of intestinal villi to well-organized structures such as the tonsils and adenoids, the appendix and pear patches which are found within the intestinal lining and contain well-defined follicles and T cell zones. Lymphoid cells are found in various regions with the lining of intestine. The outer mucosal epithelial layer contains intraepithelial lymphocytes, many of which are T cells. The lamina propria, which lies under the epithelial layer, contains large number of B cells, plasma cells, activated T cells and macrophages in loose clusters. Microscopy has revealed more than 15,000 lymphoid follicles within the intestinal lamina propria of a healthy child. Pear patches, nodules of 30 to 40 lymphoid follicles extend into the muscle layers that are just below the lamina propria. Like lymphoid follicles in other tissues, those that compose pear patches can develop into secondary follicles with germinal centers. The overall functional importance of mucosal associated lymphoid tissue in the body defense is underscored by its large population of antibody producing plasma cells whose number exceeds that of plasma cells in the spleen, lymph nodes and bone marrow combined. Friends, some cellular structures and activities are unique to the mucosal associated lymphoid tissue. For example, the epithelial cells of mucosal membranes play an important role in delivering small samples of foreign antigen from the respiratory, digestive and urinary tracts to the underlying mucosa associated lymphoid tissue. In digestive tract, specialized M cells transport antigen across the epithelium. The structures of M cell is striking. They are flattened epithelial cells lacking the microvilli that characterize the rest of the mucosal epithelium. They have a deep invagination or pocket in the basolateral plasma membrane, which is filled with a cluster of B cells, T cells and macrophages. Antigens in the intestinal lumens are endocytized into vessels that are transported from the luminal membranes to the underlying pocket membrane. The vesicles then fuse with the pocket membranes, delivering antigens to clusters of lymphocytes and antigen presenting cells, the most important of which are dendritic cells contained within the pocket. Antigen transported across the mucous membrane by M cells ultimately leads to the activation of B cells that differentiated and then secrete immunoglobulin A or antibody A. This class of antibody is concentrated in secretions and is important tool used by body to combat many types of infection at mucosal sites. So friends, this is all about the secondary lymphoid organs. If you like this video, please subscribe to our channel, press the bell icon and don't forget to share this video with your friends. Thank you.